Welcome to today's lecture. Today we'll continue to talk about humoral immune response. In last section, we have mentioned that activated B lymphocyte return to the follicle to form the germinal center. And what's going on in the germinal center? This is the germinal center. It's composed of two zones, the light zone and dark zone. In the dark zone, there are many rapidly proliferated B lymphocytes, and the light zone is composed of the activated B cells, follicular dendritic cells, and follicular helper T cells. B cells undergo several modifications in the germinal center that ensure that it produces a more effective humoral immune response. These modifications include somatic hypermutation, affinity maturation, and class switching. And finally, B lymphocyte can grow into the plasma cell or membrane B lymphocyte. Somatic, in the dark zone, the B cells rapidly proliferated, and somatic hypermutation will occur. Somatic hypermutation refers to the mutation in the variable regions of the immunoglobulin, especially the hypervariable regions, which combined with the epitope of the antigen. After somatic hypermutation, some B cells are high affinity for the antigens, while some other cells are not. And those high affinity B lymphocytes can survive, while the low affinity B lymphocyte will die in apoptosis. But what's the reason for that? In the light zone, B cells survive depend on two types of the cells, follicular dendritic cells and follicular helper T cells. And let's look at, this is the follicular dendritic cells. Follicular dendritic cells express Fc receptor and complement receptor. That means it can accumulate so many antigens in form of the antigen-antibody complex and complement complex. So there are lots of the antigens on the surface of the follicular dendritic cells. And B lymphocyte will recognize the antigens on the follicular dendritic cells. Then they will receive a survival signal. And B lymphocyte will internalize the antigen and process those antigens for recognition of the follicular T cells. Then T cells can be activated. And the B lymphocyte also can get a survival signal from the helper follicular T cells. But as you can see in this picture, this is the germinal center. We can see more than 90% of the cells are B lymphocyte, shown as in yellow. And only a small fraction of the cells are T cells, follicular T cells, shown as in blue. So that means B cells, they must compete with one another to contact with the T cells. Then they will get a survival signal. After the competition, the B lymphocyte with higher affinity for the antigens can get survived signal because they can bind with the antigen on the follicular dendritic cells and they can interact with the T cells. So they can survive. While others B lymphocyte with low affinity, they will die. This process is called affinity maturation. After this selection, those high affinity B lymphocytes, they can proliferate into the plasma cells, and then they produce the antibody, and this antibody will have a high affinity to the antigens. Another important event in the light zone is class switching. Class switching means the B lymphocyte can produce different classes of the immunoglobulin, and this process is regulated by the helper follicular T lymphocyte. T cells can express so many different kinds of the cytokines. In the presence of the different cytokines, B cells can proliferate into the plasma cell and then secrete different classes of the immunoglobulin, such as the IgG, IgA, IgE, and IgM. So that's the reason why in our body we have 5 classes of the immunoglobulin. This step is called class switching. In summary, in humoral immune response, B cells are activated by antigen and secrete antibody that can act to eliminate antigens, especially those extracellular pathogens. B cells undergo several important modifications in germinal center that produce a more effective antibody response. Affinity maturation and class switching in the germinal center require the help of follicular T lymphocyte. That's all for today. Thank you for your attention.